much, Nikolai. I guess it was a bit of a shock there. I was getting ready to have my lunch and suddenly I'm talking. So that's fine. So today I'm going to talk about some work that my group's been involved with over the past couple of decades, looking at the development of vaccines for bovine respiratory disease. And I guess what we would say here is that a lot of the people in the room are looking at saving people, and so we're working towards then once you save them, how do you feed them? So I'll go over a little bit of background about what is bovine respiratory disease, the role of the pathogens in BRD development, and then what we've been doing towards making better vaccines for BRD, and then how we go about testing those in what is quite a complex disease. Pardon me. So BRD is a multifactorial disease in that an animal getting infected with a virus doesn't necessarily mean that animal is going to go on to develop disease. There's environmental factors that go into the development of the disease, and I often describe this as it's kindergarten for cattle. So when we put our children into kindergarten, they get exposed to a whole range of new pathogens, and then they develop disease for that, or to that. So stress plays a big role in BRD, so cattle are getting moved into new environments, they're adapting to new diets, they may have been transported for long distances, particularly in Australia where that can be for days in some cases, and then they get exposed to a whole range of new pathogens that they might not have seen before. There's 4 main viruses which I'll talk about a bit about today, but then also bacteria. So we work on a BRD model of cattle first getting a viral infection that then predisposes them to a secondary bacterial infection, which is what we treat clinically as BRD. And they're most susceptible to that in that first 2 to 3 weeks when on arrival at the feedlot. So after that period of time, things tend to settle down in terms of treatment of BRD. And so how do we know that? Just quickly. So in the past few years we've been working on the epidemiology of BRD, so just not the role of the pathogens. And so my epi friends have created this causal diagram for those things that might contribute to animals getting BRD. And as you can see, it's quite complex. The pathogens do certainly play an important role, and they're on the diagram, but there's a lot of other things that also play a role in the development of the disease. And as I said, in that first 21 days is that critical time point. After that, things start to settle down, and certainly by day 50 on feed, we don't see much BRD after that. And you'll see here I have in grey the pre-feedlot area, and that is that this used to be a black box. So we didn't know, we knew there were things that influenced cattle getting BRD in that time, but up until very recently we didn't know what those things were. And so my group's been working on actually turning that from a black box into a grey box, but that's another 7-year story. So from our recent study, we've seen over a 5-year period, a study involving 35,000 cattle, and you can see here that we certainly see that peak in that first 2 to 3 weeks on feed. So that's our challenge. We need to get these cattle immune or being better able to fight these pathogens in that first 2 to 3 week period. And in terms of the actual cost to the industries that we work with, in Australia our feedlot sector is about 1 million cattle get turned off from feedlots every year, somewhere between $60 and $100 million annually. In North America, that figure is somewhere between $1 and $5 billion a year, $1 and $5 billion a year, depending on what you read and who's written it. But the industry is about 10 times larger. Now these costs are this peak, this here. So what animals are actually treated clinically? And we think that there's actually a whole range of animals that actually have BRD, but they just don't present with it and therefore don't get treated. So the cost of production is probably much higher than what we actually think they are. So who are the main players in BRD? Well, we like to think of it as the top 4 or 5 viruses. Historically, when I've been working on this since the last about 18 years, bovine parainfluenza was always included in that, but we don't see it very often. So in the study I talked about before, of 35,000 animals, 18% getting BRD, we saw or detected PI3 twice, 2 or 3 times. So it's not a big player, or at least it doesn't seem to be. What's actually come to the fore in more recently is bovine coronavirus. And so we're seeing, previously not thought to be mainly enteric, but we're seeing that in respiratory disease in cattle as well. But then there's a whole list of other viruses that if we really wanted to look hard enough, I suspect we could actually find them. So it comes down to that question about, yes, we know there's predominant viruses, but then when we bring these cattle into these environment, its exposure to those any particular virus may actually make them more likely to get disease. And then we also have the secondary bacterial infections that then is what we see clinically. Of course, there is evidence that some of these bacterial pathogens are probably primary pathogens on their own right. So hemolytica expresses a toxin which kills the immune cells, and so it could be very important in that. And Mycoplasma bovis is also one that's come to the fore more recently in the last 5 to 10 years. So a big four, as I said, we've substituted PI3 out of this team, is bovine herpes virus, and so with cattle coming into Australian feedlots, most cattle haven't seen this virus before, so it's a very big player and one that we regularly isolate from cases of BRD. The bovine viral diarrhea virus is also a very common virus in cattle, has quite a complex etiology in how it's transmitted through populations we won't go into today. And so these are the two main ones that I'll talk about today and that we've been focusing on in the last little while in my group. Respiratory syncytial virus is what we call the holy grail of Australian virology. So we know that cattle see this virus, we know they get infected with it because we can monitor that through changes in serology, but we've never been able to actually isolate the virus even though we can physically detect it, or can detect it in clinical cases, in nasal swabs, in lungs from cattle that have died from BRD. And then bovine coronavirus, as I said, is the new kid on the block. So we have a relatively unique situation with respect to BRD in Australia in that the adoption of vaccinations for that is relatively recent. So Rhinogard, which came onto the market probably about 10 years ago, I suppose now, was developed by the Primary Industries Department in Queensland and is now sold through Zoetis. Was developed specifically for the feedlot sector. So it's a live, modified live intranasal vaccine. They get one dose at induction on arrival at the feedlot, and then that gives them generates a non-specific rapid immune response, and they develop acquired immunity in half of that. Pestigard was developed for BVDV. It's not specifically targeted towards the feedlot sector. It requires 2 doses. So if you only give a dose at arrival and then you want that protection that first 3 weeks, it can be problematic. Although there is work going on to see the benefit of a single dose. Same with Bovilis, which is for Mahony hemolytica, and again developed a number of years ago and is for that primary or potentially primary bacterial pathogen. And more recently we've had inactivated Mahony hemolytica and BHV, which is IVR as well. Interestingly, in the States, where we know that BRD is a much bigger problem, they have in excess of 150 vaccines for the same disease, which have been developed over the last 15 to 20 years, and it still continues to be a major problem in that country. So we need to start doing this a little bit differently. So, as I said, and what my talk will be focused around today is our work developing live modified vaccines for BRD and other cattle applications. The advantage is we get this single dose. So on a feedlot, when the cattle come in, you like to do what you need to do to them, put them in their pens, and then leave them alone. Because the more you muck around with them and move them around, that costs you money because it takes some time off feed. They need to adapt to different conditions and that sort of thing. So once they're in, and then that's where you want to leave them. In our other, some of you will know, some of you will not, in extensive beef industries in Australia, we have the opposite problem. So our cattle could be spread over thousands or millions of square kilometres, so you don't see your cattle very often. So again, single-dose technology is going to be a very important player there. The other advantage of modified live is they follow that similar course of infection. So we know that if cattle get a natural infection in a relatively benign environment, But in most cases with these viruses, they will recover and then they will have acquired immunity. So the advantage of the live viral vaccine is that it gives you that similar mimic of what they would otherwise see in an infection, but without hopefully disease. As I said, you also get that rapid, non-specific stimulation of the innate immune system, which is very important on that first period on feed. The disadvantage, and we've heard a little bit of that today in the other parts of the conference, is immunocompetence. So we don't really want to be giving a modified live vaccine to an animal which might be severely immunocompromised. So it's a balancing act between getting protection and not causing disease. There's potential issues with recombination. So we're giving a live virus, so there's potential for that virus to recombine with other similar viruses that the animal might be infected with. And then of course, with some, such as the herpes viruses, we have the issue of latent infections, whereby once the animal is infected, it remains infected for life, although not always expressing or secreting virus. So the work that my group's been involved with over a number of years is using bovine herpes virus as a modified live vector for BRD. So as some of you may know, it's a DNA virus, and we typically consider those to be very stable, and that's certainly the case for BRD, and I'll show a little bit of evidence for that today in the work that we've done. It has a number of factors which we know are related to virulence, so if we knock those out, that we will see the reduced capacity to cause disease in the field, or in animals, sorry. But I don't think that we know all the things about that, so there's still things that this virus can do that we don't really understand. But all things considered, we think it's an ideal vaccine vector compared to the other viruses involved in BRD, which are mostly the RNA viruses. So, as I said before, we are focusing on a live attenuated vaccines. The animals receive a single dose at induction, intranasally, and then they go off into their pen. It undergoes replication, immobilises the innate system, mainly an interferon-based protection, and it gives them that key period of protection against this important virus. So the work that we've been doing is on developing it as a recombinant vector, whereby, because of the large genome, it's able to accommodate a number of insertions and deletions of genes, either for reduced virulence or the incorporation of antigens from other pathogens. It's very stable in its replication. I think there's some debate now if that's true for all the herpes viruses, but it certainly is for BHV. It's relatively easy to manipulate, and I'll talk a little bit about how we do that in my lab. And it's relatively host-specific, because nothing's ever truly host-specific. And it also has the potential for use as a marker vaccine, so whereby you can differentiate infected from vaccinated animals, but I won't talk about that today. So what we've been doing is looking at ways in which we can actually take antigens from one particular virus. We have the advantage that for a lot of our viruses, the major neutralising epitopes are known. So if we can take those out, or antigens and epitopes, and put them into our bovine herpes virus vector, then generating multifaceted immunity. Our first target, as I said before, was using BHV as a vector to carry BVDV antigens. But we've also used a system we've developed in our lab, which is an infectious clone system, to generate libraries of mutant genes, so whereby we can better understand the biology of the virus. So what we do is, if you like, here we have the bovine herpes virus genome, so a DNA virus that we've cloned as a bacterial artificial chromosome to allow us to grow and manipulate it in bacterial cells. We then, in this case, we want to target the glycoprotein E, a non-essential gene within the virus, so it doesn't need it to grow in the lab or in animals. And we are replacing it with an antibiotic resistance cassette in this case. We can either generate that through PCR or using transposition. We target that into the GE locus in the genome. We then double select for one which is for the infectious clone, the other for our transgene. Those colonies which are carinamycin resistant, we isolate the DNA back from those colonies, transfect it into bovine cells, and then we have our recombinant virus, which is, in this case, has had GE knocked out. So we've used the same process to put in antigen genes or antigen expression cassettes from other viruses. This is a map from one of my PhD students, and what we did here was we used transposition to make random insertions into that infectious clone. And those genes that are marked in red are those genes that the virus absolutely requires to grow in the lab. So we know that if we knock out these genes, that the virus doesn't grow anymore, so when we're putting in or making deletions, we don't touch these genes. We focus on those ones which are shown in black. So, as I said before, our main target was bovine viral diarrhea virus, or BVDV. As I said before, it's an RNA virus. It has a single open reading frame that it translates as polypeptide from, which is then cleaved into the various proteins that make up the virus. So here we have the major immunological determinants, and the primary one of that is E2, and so that's the one that I'll focus on today. So we've taken this gene, and we're putting that into VHV. The first issue that we come up against is the bovine herpes virus, as before, it's a DNA virus. BVDV is an RNA virus. They replicate in different parts of the cell. And as a consequence of that, if you just, for some of BVDV genes, if you take those and put them in directly into the nucleus, then they're actually affected by cellular mechanisms that they don't usually see, such as splicing. And they're also not subject usually to mRNA transport, so if they are produced in the nucleus as a single thing, then they will actually stay there, and so you don't get protein production. So BHV is also a high GC virus, and BVDV is a high AT virus, and so they actually have different codon usages in their genes, and this is a common mechanism in viruses to control protein expression. Oops. And this is all even though they infect the same host via the same pathways, but they do probably replicate in different cells. So our main concern in taking a gene from BVDV with the RNA virus and putting it in BHV is the presence of cryptic splice sites. So because it's in the nucleus, it's now subject to RNA splicing, which it's evolved not to have to deal with. And so we look for consensus donor and acceptor sites, but the problem with that is that they can sometimes be dependent on the methodology you use. And so one of the key examples of this was shown for classical swine fever virus, which is a relation of BVDV. And what the authors were able to show was if they take those genes as is and put them into their expression vectors, that this splicing actually does occur. And it's also been shown for other viruses, such as equine infectious anemia virus. There's been anecdotal reports that it's a common problem in all BVDV and other RNA viruses, such as BRSV. But there was a study published a few years ago now on a separate strain of BVDV, and what they showed was that sometimes it's true and sometimes it's not. So if they made what they thought were the cryptic splice sites, took them out, and they showed that they got expression of both the synthetic and the genomic version. So it seems that if you want to change your gene, then you need to actually determine it empirically. And as I said before, because of the differences in GC and AT content of the two viruses, they also do use different codons, a different codon bias. So they'll use some codons in preference to others, and that's certainly the case. And you can see here, it's well documented in the literature that for particular viruses, this actually does occur. So when we're overexpressing antigen genes, we need to take that into account. So our solution was to make, as I indicated before, a synthetic version of the E2 gene, and then try and take out as many of these RNA control units, or consensus signals, as signals that we thought might be present. Site-directed mutagenesis was a problem, but when we looked at it, there was a lot of different things we'd have to change, and so we went on the synthetic gene approach. And when we did that work, this was actually quite an undertaking, whereas these days we just ring up LifeTech or some other gene script and get them to make us 1,000 different versions. So we removed the cryptic splice sites, we changed the codon used to make a high GC virus. We wanted it to look more like a BHV gene because it was our vector. About 1 in 10 bases, they required changing about 1 in 10 bases. And we also had to add on a signal sequence because it's normally produced as a polyprotein, so there's only one signal sequence within the BVDV genome. And we ended up with our prototype vaccine, and we showed expression of the E2 cassette, put it into VHV using that methodology I talked about before. Our bovine herpes virus can now be neutralised by BVDV positive sera from cattle, so that gave us good confidence that we had it in the right structure and everything in the virus. So we then came to the end game, if you like, of the situation. That was, how do we test efficacy? So we just infect some cows and look at antibody responses and look at the normal factors we'd look at, such as the duration and the quantity of virus shedding. The problem with BRD, because it is a multifactorial disease, when we challenge with some of these viruses, we don't necessarily see severe or even overt clinical signs because they're not in that feedlot environment. So what we actually do is we use a dual challenge model. So what we do is we vaccinate, and then within our peak BRD period, we then give them a challenge strain. In this case, we're looking at the BHV side of things, and so we're giving them a challenge strain of BHV. And we monitor them for temperature and swabs and clinical signs. And then about 4 or 5 days after that, we give them Mahony hemolytica to simulate that secondary bacterial infection, and then do similar monitoring of them. So then we monitor our cows for these clinical signs. Now, when you can do this with your nose, when you're looking for nasal discharge, it's actually a bit problematic. But we do see down here a little bit of nasal discharge. So I get what I would normally describe as a mild cold. So they're certainly not, if we don't want to induce actual field clinical BRD, because I don't think we would be or should be allowed to under animal ethics. But they get a mild cold. With these animals here, in this trial we saw a little bit of lethargy, so they're a little bit non-responsive when you had to get them out and put them into crush. So it's actually a mild cold sort of thing, as opposed to clinical BRD, which would actually place them at risk. And you can see here, this work was done a little while ago, so we've got our recombinant freeze-dried vaccine here. These are our unvaccinated controls, and we've shown pretty good protection. These are total clinical scores. This is the commercial vaccine that's available for BHV, and again, it's equivalent to what we've seen before. But as you can see here, we had a problem with our BVDV side of things in that we don't see any escalation in the clinical signs in these animals. And so that was one thing. We also had problems detecting the BVDV virus in nasal swabs, and so we're a bit concerned that the animals didn't actually get infected. And this is and so we then started to think about that, and so then this is a study from using a subunit BVD vaccine. And you can see here the authors have used the presence of virus in nasal swabs and buffy coat as a measure of protection. And in a conventional vaccine, they detect the virus only once in nasal swabs after about 11 days. In their treatment, so this is their novel formulations, they see virus here once. And in their control, so unvaccinated challenged animals, they see the virus sporadically, and this is fairly common in the BVDV vaccine literature. And I guess, I don't know about you, but I'm not really convinced. And so we thought, well, is it an issue with the strain of BVDV? And perhaps in the animals that we used here, our strain that we'd used wasn't very efficient in infecting cattle. So late last year we conducted a trial whereby we took 5 strains of BVDV and infected cattle with them to see if we could identify a strain that would be more suitable for use in our vaccination trials. And as you can see here, I guess the one thing is, it is very variable. So this is the strain we used in our first trial. So we see the virus every now and again, pardon me. So this is only in nasal swabs. This virus here, which was highly recommended to us, we hardly see anything at all. But 3 and 5 were actually very consistent, or relatively consistent for BVDV. And these asterisks show where we've detected the virus in serum as well as in nasal swabs. So we then went ahead and repeated some of those vaccination trials with this strain 5 on the basis that it was detected in nasal swabs for a more continuous period. And again, excuse me, we had our BHV vaccinated and control groups, and again we saw very good clinical protections, very similar to what we saw the previous time. A little bit depressing, given that this was a few $100,000 later and a couple of years, that we saw a very similar situation in our BVDV control group. So I guess at this stage, I was thinking of that maybe I need to find another career and do something else. Just before I get on to a potential explanation for that, we also looked at the recovery of hemolytica in this, and you can see there we see good distinction there. So the BVDV challenge component of it was we didn't see an overt clinical distinction. I guess that's the bottom line. Some animals were virus positive. All animals seroconverted at the end of the trial, and we didn't see that in our first trial, so we think the strain was certainly better. But when we actually looked at in close detail at the cells in the immunes in the blood of these animals, pardon me, we did see distinction between the vaccinated and the controls, and there was some benefit for those vaccinated animals. So we'll skip back there on a time. So in going over that, what we think is that in the BVDV side of things, the protection is more subtle, and so we think that because of that, it's subtle and more variable, and so we probably need to repeat those trials with more animals to try and to tease that out and or detect or look at more strains of BVDV to see if we actually do have the right one, even though we think we do. And I'll just close off by saying that perhaps our biggest challenge will be that of hopefully being the first group to undergo the registration and commercialisation of a genetically modified organism in Australia for use in cattle. Homemade one anyway. So in dealing with that, we have to deal with this perception of GMO, and it doesn't always get a lot of good press. We think that we have industry on side, and so most of our work has been funded by industry. They're very supportive, they see the need for that, they understand their obligations from an animal health and welfare point of view, that they need to do the best that they can for their animals in their care during that period. Perhaps what we don't really know is the public and consumer perceptions of actually having a GMO vaccinated animal in the food chain. There are some schools of thought that I have read that in this case, where the GMO is being used to benefit animal health and animal welfare, that may be more acceptable compared to what could you do? You make a plant that is resistant to something that you want to sell, if you were cynical. So that there is actually a true benefit there for the animals involved in this. We think that we'll get regulatory acceptance, so we've done some of these experiments under the guidance, in inverted commas, of the Office of the Gene Technology Regulator. And I guess while that was a very long process, so 2-year process, they were very reasonable. So if we knew the answer to, if there was an answer in the scientific literature, they would actually accept that. If there wasn't, they gave us a requirement to actually determine that. So they gave us the opportunity to go ahead. So BRD remains a big issue. Vaccines, I guess we're all true believers in this next couple of days. We think that that does provide an opportunity and a possible solution. In our hands, we think recombinant vaccines are probably a good way to go. But I think one thing we do need to do, and I think that some of the speakers have already alluded to that in some of our talks today, is we need to understand these pathogens better. So one thing, in animal areas, we can go straight into phase 3 and phase 4 clinical trials, but we don't necessarily understand when things don't work, why they don't work. So with our BVDV vaccines, there's probably a lot of stuff we still don't know about one of the most well-studied cattle viruses, and that is BVDV. And I'll just close off in thanking the people in my lab over the years and the funders of the work. Thank you very much.